Dah dapat kan dekat air taklim? Alright, that particular manual is I think it's about transformation lah. So partly related with genetics juga, alright, on how you guys can transform certain organism. So by using the methods yang akan disediakan nanti lah, alright. But for today, we will talk about the slightly different things lah, alright. So we will talk about these things: alleles, transposon, epistasis, and certain mutation lah, alright. Bear in mind juga sebenarnya I'm planning to give you some assignment today, alright. It's related with previous week's punya chapter, alright, regarding genetic linkage, alright. It's all about crossing over genetic linkage dengan mapping lah, alright. So I hope you guys can get prepared on that particular assignment. The duration for assignments saya akan letak di dalam air taklim. So please away dekat air taklim punya Windows lah. Alright. So dan regarding this particular topic juga, we'll get you guys to prepare some presentations, alright, for this particular topic. Alright. So I give you about 3 to 4 weeks lah for the preparation for presentation. Alright. So I will inform it later, and I will share it in air taklim juga next week lah, bukan this week lah. Alright, beginning topics. Alright, so let's talk about alleles. Do you guys familiar with alleles sebenarnya? Alright, so we talked about alleles last week, so I'm quite surprised lah no one asked about alleles actually. Alright, let me explain about alleles punya definition a little bit lah. And I guess we talk about this particular things related with genes, betul tak? Alright, last week we talk about genes. So what happened in genes adalah genes is a sequence, a DNA sequence yang akan translate into certain protein and will be expressed as a phenotypes. Right? So genes in particular is a region in the DNA lah, where they can control specific traits. Right? For example, macam shape, right? Ataupun colours, ataupun the flower colours and so on. So this is the thing yang kita bincang last week lah. Alright? So there is particular genes. Right? He talks about shape, talk about colours, maybe talk about shape of yellow, for example, right? Shape of eyes, something like that, right? Related with that. But however, allele is a different version or alternative form of the particular genes, right? This is allele, right? Genes are related with this particular colours. Tapi contohnya kita ada genes untuk colour putih, for example, bunga colour putih, right? This particular genes, this is white colour flowers being controlled by this particular genes ataupun the DNA sequence lah, right? Tapi the other alternative form of that, maybe kita jumpa the similar flowers but they have different colours, right? Maybe the colour dia colour purple, for example, right? Like Mendel did during the observation lah, dia tengok, right? This is where differentiate between genes lah, right? So that's why alleles ni dia ada banyak, right? The different version of the genes is alleles, right? Hold on. Okay, so this is the thing, alright. So hopefully everyone understand lah what allele is right now, alright. So dia adalah alternative form ataupun different version of the gene yang kita tengok, alright. Tapi I want to highlight here since you learn about genetics, right? So first of all, kita faham, right? We understand one particular traits such as colours of flowers ke, shape of the leaf ke, alright? Ataupun any other traits, rambut kerinting ke, rambut lurus and so on, there is traits, right? So this one genes can control one particular traits, right? Tetapi, alright, there are certain situation, right? Right? There is more genes that control one particular traits, right? There is more genes that can control particular traits. I give you example, right? Contohnya kita jumpa pokok yang boleh survive bila keadaan panas. Pokok-pokok lain dia tak survive, tapi pokok ni dia boleh survive, right? So what happened with this particular pokok sebenarnya, right? I mean panas kira macam kemarau lah, alright, drought, alright. So tak ada air sangat, tapi tiba-tiba one particular pokok ni dia boleh survive this particular condition. So what happened sebenarnya adalah, so they are being studied and being issued, right? So we call it quantitative traits, if not mistaken, right? So dalam dunia ataupun dalam breeding punya situation lah ya. Kalau contohnya we go for apa nama molecular breeding, right? Kita nak breed certain traits yang sangat-sangat diperlukan ataupun sangat-sangat berguna lah. Macam saya cakap tadi lah, dalam keadaan yang low water, certain plant can survive. So kita nak tengok genes apa yang evolve sebenarnya, right? So this been studied, right? Several genes, right? Yang macam ni, several genes ni yang encode protein ni, right? Dia ada several genes yang evolve untuk jadikan that particular traits, alright? So certain genes yang evolve untuk jadikan that particular traits, right? But don't worry, alright? Normally we understand lah, certain genes dia akan keluarkan certain traits, alright? Tapi under certain condition, alright? Certain condition yang it's sebenarnya kalau fix lah, contohnya colors dia tak ada variation. Colors dia akan fix. Okay, color hijau for this particular gene, color kuning for this particular genes, alright? Tapi kalau things yang can be quantified, contohnya dalam keadaan yang tadi drought tadi, alright? So they can be quantified. Selama mana dia boleh tahan drought tu, itu adalah certain several genes yang akan evolve. Right, but don't worry, kita akan go one by one. Just I want to highlight dekat sini lah, just as an introduction for you guys. First of all, kita faham one genes can control one particular traits. However, there is a condition that several genes are required to control one particular traits. Alright, so I want you guys to understand that lah. Alright, so dia ada special condition lah. Dia normally memang one traits be controlled by one particular genes lah. Right? So next kita talk about the dominance lah. Okay, I think everyone knows about dominance dengan recessive lah. So dominant dia akan mask recessive punya apa nama expression, and recessive pula dia adalah the one that being masked by the dominant punya alleles. Alright. So any questions from here? Boleh faham ke? Alright. Yeah.
Yeah.
Good. So it goes for the next one, transposon. And we talk about transposon a little bit last 2 weeks, alright? In the first week juga kita ada bincang sikit from Barbara McClintock, right? Dia jumpa these particular elements, alright? In the DNA sequence, like we talked about last week lah, alright? There's a lot of DNA sequence yang ada encode genes dekat promoter yang kita boleh transcribe, yang kita akan tukarkan dia kepada messenger RNA, and which the messenger RNA will be translate for protein punya production. Right? And this particular protein tu yang akan express things. Right? Since you guys already understand about the DNA sequence, right? So the transposon element ini adalah dia berlaku dekat dalam DNA sequence. Right? So the certain DNA sequence ni boleh melompat ataupun they can be transferred from one sequence to another sequence. Right? This is transposon. Right? A segment of DNA that are able to move within the genome known as a jumping genes. Right? So for example, this is one particular sequence. Right? So this sequence can transfer to this particular sequence. Alright? What happens since you guys learn in last week about transcription process dengan translation process. Right? When one particular region being transferred to another one, so maknanya the whole sequence ini mungkin akan berubah juga. Alright? Contohnya sequence yang asal dia akan keluar certain protein. Alright? Dia akan keluar certain protein yang involved with pergerakan akar, contoh. Tapi once kita transfer, once dia akan ditransfer ataupun dia transfer the other way around, right? Mungkin function of the protein will be changed, right? Due to the changes of the sequence yang dah berubah kat dalam, right? So this is quite important, but this is why the discovery by the Barbara McClintock ni quite interesting, sebab when there are changes in the sequence of DNA, right? There are possibilities that the sequence of DNA will encode different type of protein ataupun maybe different type of sequence yang akan encode different type of protein yang berbeza. Alright? So that's why bila ada perubahan ni, kemungkinan perubahan yang sedikit akan merubah the function of protein tu quite significant. Alright? So this is where the changes is quite important lah. That's why they are studying about this particular process. when there are changes, what happened to the new sequence yang berubah. Right? Move from one genomic location to another by copy and cut ataupun copy and paste punya mechanism. Right? Kalau copy and cut ni, right, cut will be okay if you ask my opinion. Right? Copy and cut ni dia berubah kat tempat lain, which mean that it's quite okay. Sebab for example, let's say you have one genome sequence yang size ni adalah 1000 base pair, right? 1000 base pair. Alright, and then when you cut certain region dan masukkan balik ke certain region, the sequence will be still 1000 base pair. Alright, 1000 base pair juga. Awak cut 20 sequence, alright, awak masuk balik 20 sequence kat di location yang berbeza. Alright, but it's still in 1000 base pair. Alright, however, it's copy and paste mechanism, right? Dia copy dan dia paste ke tempat yang baru, which means sooner or later, right, they will increase the genome size, right? So when they increase, this is where it turns into quite significant changes, right? So banyak benda yang berlaku bila kita increasekan dia punya DNA sequence. So there's a lot of more protein yang akan berlaku ataupun being translated inside that particular organism, right? So this is where the study goes, right? So there are two types of transposon, right? Retrotransposon, we call it class 1. Usually a duplicate and reintegrate themselves masuk lah, copy and paste. Right, this is retrotransposon. But for DNA transposon, usually it will be excise themselves and move to another location, cut and paste. Alright, so they cut, they paste kat tempat lain. But copy and paste will be a little bit more quite significant punya changes lah. Alright, so as I did mention, 1000 base pair, they may increase if the process still occurs, right? Maybe we will double up the size of the genome, maybe in 2000 base pair, for example. So this is where the study is being quite significantly being studied lah. Alright. So what happened actually? Sorry, nanti saya bagi slide mungkin susah nampak sikit, kurang jelas sebab gambar dia tak nampak sangat. Alright. What happened in retrotransposon? Retrotransposon ni tadi adalah copy and paste. Alright. So what happened in retrotransposon? Kita tengok sebelah sini lah ya. Alright. Kita ada DNA sequence. Alright. Kita ada RNA polymerase. Alright. So dia ada RNA polymerase yang akan copy this particular region menjadi RNA. Remember RNA polymerase that is required for transcription process. Alright, so that's why the result will be RNA intermediary lah. There will be ada mRNA yang akan dihasilkan. What happened with this particular RNA? Alright, they will be reverse transcriptase. Alright, reverse transcriptase ni means that dia ada RNA, dia reverse transcribe this particular RNA to become DNA balik. Alright. So when they have this particular DNA balik, right, dia akan cari target site. Alright. Dia jumpa target site dan dia akan masuk ke target site untuk dimasukkan ke dalam. Right. So this is what happened. Right. Copy and paste lah. Masukkan ke target site. So this is where the target site is. Kita akan bawa pasal target site lah. Kat sini. Alright. The target site. Alright. So that's why in retrotransposon, right, they have the RNA intermediary ke dalam, right, retrotransposon. So they copy and paste punya mechanism. They copy this particular region, right, so they reverse transcribe dia jadi another DNA sequence, right, this particular DNA sequence akan masuk ke that particular region, right. So this is what happened. Alright, for DNA transposon pula, there is very ni lah, sama je. DNA transposon is a very straightforward punya proses lah. Alright, what happened? Di sini will be cut, right? It will be cut and excise, right? So this particular region akan masuk ke dalam this particular target sequence. Itu saja. Dia cut dan dia ada ready untuk masukkan ke dalam this particular sequence. Alright, so that's why I said that when it cut and paste, it will be more straightforward. So it just more on just removing one region to another region, right? But for retrotransposon, dia copy this region, dia buat DNA baru dan dia masukkan ke dalam this particular region, right? So which means dekat sini tak ada hilang, dia just copy saja, right? Same goes with this. Different story with the DNA transposon. Dia cut this particular orange region, right? Dia akan masuk ke dalam sini, right? So di cut region ni dia dah akan ligate balik, dia akan bercantum balik lah. Right, so this particular region terkeluar, dia pergi cari target site dan dia akan masuk ke dalam this particular target site. Hilang dan masuk balik. So which mean that dia punya DNA sequence tu, for example, if 1000 base pair, right, they still remain 1000 base pair juga. But for this particular sequence, right, dia ada region, right, so region ni akan di copy by using the RNA intermediary lah. So what happen, this copy dia akan di reverse balik jadi DNA. So they will be bind to this particular region. So sini tak hilang, tapi dekat sini bertambah. So this is the thing yang I did mention lah. Right, if this particular process occurs frequently, right, it might occurs ataupun alter the DNA sequence yang ada dekat dalam tu menjadi semakin banyak. So what happen adalah bila DNA sequence semakin banyak, there's a lot of protein will be processed. So we need to understand lah what type of protein yang dihasilkan and what type of the function of the protein yang akan hasil. Alright, so this is the thing. Tapi kalau let's say protein tu banyak digunakan, for example in human situation lah ya. Kalau protein banyak dihasilkan, but this particular protein is not really have some useful function dekat dalam human bodies, right? So it can be removed. However, if it occurs frequently, right, it may cause cancerous punya situation. So this is quite bad for the medical punya situation. So that's why they are under studies. So that's why Barbara McClintock ni they receive the awards under the medical punya studies lah. Right? So I just want to highlight both class of transposon is can become autonomous or non-autonomous. Autonomous transposon can be moved by themselves, right? But non-autonomous adalah they require some presence of another transposon to move. Alright? So for example macam DNA transposon ni lah. Dia ada transposon lagi so to move ni, alright, to move ni sebenarnya adalah when they have another transposon punya region element kat sini. Alright? For example this one. I give you example macam DNA transposon. Alright? DNA transposon dia ada transposon element kat sini, right? So when they want to move, they being triggered by this particular region untuk masuk. So this is the region yang the transposon region ataupun element yang saya cakap tadi lah. Alright? So they require this. For this one, they just need required another region yang dia perlu pindah. But for this one, they are already they are ready to move. So this is the thing yang saya nak cakap lah. Alright, non-autonomous transposon require the presence of another transposon to move. Alright, ini adalah transposon sebab they can move. But normal DNA, alright, they will not move lah unless they have this particular element that trigger them. Alright, so this is the region of transposon dia akan boleh berkeluar dan pindah dan masuk. Dia macam give you example macam remote TV lah. Kalau dia ada battery slot dekat dalam, alright. So when you want to remove the battery slot, you can put them another battery masuk kat dalam. This is the element lah yang saya cakap lah. But they ada required of pertukaran battery, okay, dia masuk ke dalam, alright. But for retrotransposon ni, alright, they can move by themselves, alright. So they just find region ni, so they need just a space untuk dia masuk. But this one dia ada transposon yang asal, region yang asal, dia akan boleh berpindah. So this is the thing yang saya nak cakap lah, right, transposon features ni. Dia ada IS element. This particular element, so after being studied, right, there's biomedical study, furthermore study that been studied by many people, right. So they found this particular element, right. So the insertion sequence is the insertion sequence elements, the main part of the transport element that can move from one location to another location. Alright, so they have this particular region, alright, insertion element, protein coding kat dalam ni, the inverted region, right? They found this particular region, right? Whenever transposon move, they have this particular region, right? So what happened in this particular region? They are the transposase enzyme responsible for making the cut in the chromosome region of which IS element can be inserted, right? The inverted terminal repeat ITR for the transposition and recognition site for the binding of the transposase. Ini kena ada, dia akan masuk, dia akan tolong cut this particular region. Dan this particular region boleh dimasukkan of the DNA barulah. Alright. So for example, this one tadi, right? DNA transposon tadi, right? So they found that there are the IS element in between of this particular region. So they will be removed, will be cut, and then they can like get to another place. Right, so this is the thing macam cakap, non-autonomous transposon required presence of another transposon to move. They will move to another place. Right, so let's go this one. Right, they found this particular IS element, right, insertion sequence element. Right, so this insertion sequence element ni dia ada transposes yang akan tolong cut dan dia akan membantu untuk masukkan ke dalam this region, right, particular region, the sequence baru. Alright, so this one, the inverted terminal repeat ni menjadi penanda lah. So whenever they have this particular inverted repeat, right, 5 to 11 base pair ataupun 50 base pair ni, right, this particular dark pink dengan the color black ni, right. So whenever they have this particular region, right, they knows, right, they knows in this particular DNA sequence, whenever this particular inverted repeat ni exist, there might be a possibility of transposon can occur kat sini. Right, so this is what they happen to know. Alright, first of all, they found that the genes can jump. Alright, so that's why when the genes can jump, they can detect. So apa yang berlaku? Kebanyakan gene ni melompat apabila ada this particular insertion sequence punya elements. Alright, so when they have this one, they will change and transfer them to in different region lah. Alright, so any questions dekat sini? Boleh faham ke? Boleh faham ke? Boleh. So don't worry about this is just another ideas ataupun knowledge lah for you guys will understand about this particular DNA sequence lah. This is one of the element yang ada dalam DNA sequence yang quite unique. If you ask me, I think it's quite unique as well. Sebab for me, if I learn about DNA sequence, I never thought about there are some genes that can transfer to one place to another place. Alright, to be logic lah. Sebab fikir balik kenapa genes boleh bertukar. Alright, sebab they already have that particular region yang dia akan di-transcribe dan di-translate. Unless dia akan jadi mRNA lah. Kalau mRNA kita tahulah, mRNA akan dipotong sebab dia certain introns dengan exon punya elemen yang kita akan buang untuk masukkan ke dalam. Alright, so next kita go for epistasis. Alright, laju sangat ke?
Okay dah, Doktor.
Alright, okay. So don't worry, after epistasis ni, I will just give a break lah and then we go for the mutation. Alright, so mutation ni there's several things yang I need to you guys to understand a little bit lah. But they quite interesting if you ask me. Right, epistasis is a condition sebenarnya. Right, epistasis is condition ataupun phenomenon ataupun situasi. Right, so let's talk about epistasis. What actually epistasis ni? A relationship between genes where an allele of one gene, height or mass, the visible output or phenotype of another genes. Macam susah nak faham sikit kan? But don't worry, I give you example here. Later on I will explain lah. Genes with epistasis relationship tend to code for protein that exhibit on particular phenotypes. Right? So this one is the key there. So let's see apa yang berlaku kat sini. Okay. Buat dekat sikit lah. Alright. So macam saya cakap tadi, alright, there's certain genes, alright, ataupun there's certain traits, alright, they will be encoded by the certain genes. Remember, macam tadi cakap ni, pergerakan ataupun yang kadar boleh pokok survive dekat dalam keadaan kemarau, for example. Alright, so what happen dekat sini adalah there are few genes yang involved. Alright, there are few genes that involved to make one particular trait, for example. So, ini contoh saja, ini contoh saja. It's not especially exactly what will happen lah, but this is just example. Alright. So in normal process, right, in order to have this particular output oval, bulat dan color biru, right, they required 3 particular genes involved, for example, right. First kali genes yang akan ada white paint. So dia akan sediakan base color putih lah. Alright. There are certain genes that involve for adding the blue color. And one genes that involve akan menyebabkan the circle color ni akan keluar. Right? So this is a relationship between one genes to another genes. Right? One gene to another genes. What happen if one genes is defect? Right? A ni function betul, B ni tak function betul, tapi C ni still function betul. Ataupun let's see this one. A is broken. Right? A is broken. Dia tak boleh supply the base color, the white color. Tapi B ni dia boleh still add the blue color, still function well. And C ni dia still function well untuk draw the circle color. The circle shape. Alright. Only A gene is defect. Tapi it will affect the output lah dekat ujung. Right? So which means that a relationship between genes where one allele of genes hide or mask the visible output of the phenotypes. Alright? So this relationship of between genes lah. Right? Let's say A ni defect. A macam biasa ni A dia provide the best color of white lah. Dan B ni added the blue colors, and then C dia akan buat circle color. So this is the relationship between them. Alright? But however, if one in the relationship is broken, right? Right? So maybe let's say blue is broken, right? Blue is broken, A is still good, right? A is good, B is broken, and C still can draw. The output might be, there will be the white color circle. Still can show the output, but not as what we expect of the output lah. Right? So kita tengok, this is the relationship. So this phenomenon is called epistasis. The series of genes, right? They involve in one particular traits ataupun one particular phenotype yang kita nak tengok. Alright? So they are being masked ataupun they are being some change due to the unfunctional of one particular genes involved. Alright? So nanti kita akan go through for the mutation. So mutation is quite interesting. Alright? Mutation boleh ganggu one of the genes lah. Alright? Bila mutation ganggu one of the gene, it will go like this lah. Right? It will go like this. Macam tadi juga contohnya kita ada pokok yang survive dekat dalam keadaan yang kurang air ataupun drought condition. Kurang air. There might be one gene kat dalam tu dah diganggu. Menyebabkan dia boleh survive. Right? Compared to pokok-pokok spesies yang sama, pokok lain tu semua mati. Right? So there will be one gene yang involved kat situ yang terganggu menyebabkan dia boleh survive in that particular condition. Right? So this is epistasis punya situation ataupun condition. Right? So for me it's very very interesting. Right? Because it widen my view ataupun you guys punya view lah. It's not just recessive gene yang being masked by the dominant. Right? So we need to know in detail what is the actual situation. Right? First of all, kalau kita tengok one particular situation ataupun phenotype yang kita nampak tu, first of all, think in mind, or maybe this is dominant atau maybe this is recessive lah. Alright, so if we go further and dig further, we might find the certain situation is in epistasis condition. Alright, so kita tengok, oh sebenarnya there's a lot of gene involved. This particular gene is unfunctional. Alright, so due to that, so they affect the expression of the downstream of that particular expression. Right, so just imagine ni adalah one protein, another protein, and this another protein yang involved together, dia tunjukkan benda ni. Right, so that is a epistasis punya situation. Alright, so we talk about the mutation. Let's have a quick 2 minutes break. Alright, so we will continue back afterwards. Alright, maybe inshallah another 20 to 25 minutes, then we done for today's session. Alright, so question while you guys are resting. Alright, in the central dogma, dekat mana mutation ni occurs sebenarnya. Alright, mutation ni dia akan ganggu protein function lah. Alright, kat mana sebenarnya mutation ni heavily impact this particular situation. Alright, so I give you guys a break and try to think of it. I will come back around maybe 10:37 later on. Alright, so have a good break first. Alright, you guys ready? Can we continue?
Yes.
Alright. So I asked a question just now. So anyone want to answer? Just your opinion lah. Adakah dia berlaku dekat DNA ke? Adakah dia berlaku dekat mRNA ke? Adakah dia berlaku dekat transcription ataupun translation ke? Alright. So actually dekat mana mutation bila dia berlaku, dia akan memang affect heavily towards this particular process lah, centrodogma. Anyone want to answer? Tak saya petik nama lagi. Right? Anyone want to answer?
DNA ke?
DNA. Dekat DNA siapa tu? Sorry. Nama? Shu. Alright. Dekat DNA. Maksud awak berlaku dekat asalnya. Betul tak? Dekat core dia. Kalau berubah dekat sini, everything will change. Alright? Betul ke? Macam tu maksud awak?
Ya, kat situ DNA tu.
Alright. So actually that is the correct answer. Right? Bila kita tukar dia, right? Bila kita tukar dekat sorry, jadi saya go balik kat situ. Bila kita tukar dekat sini, right? Dia akan ganggu the whole process. Alright? So which means similar macam photocopy machine lah. Alright? Kalau dekat asalnya awak dah silap, towards the end tu memang akan hasilnya silap lah. Right? Give you example macam ni lah. Right? This is a cell sickle disease lah. Right? Normal red blood cell, this is hemoglobin. This is this particular shape, the platelet shape lah. Right? Sickle cell genes, dia bentuk sickles. Right? So this is what the situation happen lah. When a normal blood pellet is there, right? Dia tak ganggu the whole process of the blood flows lah. However, is the sickle genes appears. So what happened? They will change the shape of the red blood cell. So what happened? They will clot in certain situation. Alright. So they mengganggu the blood flow of the process inside the human body. Alright. So what happened dekat sini sebenarnya? The changes are only one amino acid sahaja. Alright. Disebabkan oleh perubahan one amino acid. Alright. From glutamate to kepada valine. Right, asalnya glutamate, tapi when it change to valine, right, they change the shape is there, right? Just imagine what happen if a lot of changes in amino acid ni berubah, right? So one changes can affect this particular situation. What happen bila dia ada banyak amino acid yang berubah, alright, ditukarkan. So this is the thing yang saya tanya tadi, alright. So what happen adalah dia change dekat one sequence, dia berubah dekat sini. So this is the thing. Alright. DNA dekat sini. Alright. Mistake dekat DNA. CTC ni, it will encode for CTC. Remember last time, the codon that we talk about during the translation process. Alright. The CTC ni will encode. Alright. CAC. Alright. So bila ni DNA ni di transcribe jadi CAC. And CAC ni pula akan ditranslate kepada glutamate punya protein. Alright. What happened adalah change dekat sini. Perubahan dekat one amino acid dekat sini. CTC ni jadi GUG. Alright. So dia akan encode the valine punya sequence. Right. This one changes in one amino acid in the blood pellet punya formation. Right, so it caused a severe situation. Alright, due to the changes of the amino acids on one particular function. Alright, so mutation at DNA level, right? Mutation at DNA sequence will lead to error in the initial transcription process. Strand and the protein will be translated. Alright, perubahan dekat sini affects this particular process. Alright. So inilah yang kita kena faham lah. Tapi undeniably bila DNA sequence ni elok, ada juga yang berlaku dekat mRNA, ada juga yang berlaku dekat protein, alright, ada juga yang berlaku dekat kromosom juga. So memang ada. Tapi apa yang saya nak highlight kat sini adalah the severity of the changes, alright, the severity of the changes when it change in initial phase, alright. Initial phase ni dah change. So they akan encode the whole thing downstream into the one that are incorrectly being copied lah. Right? So mutations, right? Right, there's a lot type of mutations, right? And the types of mutation they affect DNA level, protein level, chromosome level, right? So source of mutation, it inherited, all right? It inherited, all right? Maybe the cell sickle gene, if I'm not mistaken, is a inherited disease, right? Inherited disease where people dia akan nampak dia punya streamline on that particular inheritance of the existence of this particular cytogenes, right? So ataupun dia akan berlaku spontaneous, right? Into the central dogma replication yang kita sebut lah, either in the replication, in transcription process ataupun in translation process, ataupun dia berlaku with the environmental factors, exposed with mutagens, exposed with high UV intensity, like terdedah kepada cahaya matahari yang sangat-sangat lama, it can change ataupun somehow will damage your skin ataupun sel juga sebenarnya. Gamma radiation of course lah, dekat x-ray and so on, right? So prolonged exposure. Same goes with dalam laboratory jugalah, right? We would like to highlight in our laboratory kita ada staining, right? Staining untuk kita punya gel untuk tengok DNA, right? Dekat situ ada mutagen yang kita pakai untuk highlightkan gene yang kita DNA yang kita ada, alright? So please be careful when you are in the laboratory later, alright, as well. So just please always wear glove and covered shoes. By Dr. Garina did mention covered shoes as well, alright. Jangan pakai lab coat lah. So ini yang precaution yang kita selalu guna lah untuk pakai. So please do take care of yourself later. Okay. So the type of mutation. Oh, by the way, when you talk about sickle gene inheritance and so on, alright. So there's a lot of I think nowadays when you want to get married and so on, they also checking a few things lah. I think blood type, blood races and so on, right? Races negative and so on. So ini benda-benda yang medically dia akan check supaya bukan nak kata compatible or not compatible, tapi they just want to highlight so this is what happened with this being exposed. Contoh races negative, with races negative it might be effects with the the offspring later on and so on, alright? So this the thing yang biasanya orang akan buat checking lah nowadays before kahwin and so on, alright? So this type of thing yang bukan nak kata mutation, yang saya nak cakap adalah inheritance lah, alright? So dia akan ada checking before on, dia akan tengok lah you guys get married. I think apart from the HIV test and so on before getting married, there are a few other tests kadang-kadang dibuat lah nak tengok the medical history and so on, alright? So extra things that you guys extra knowledge for you guys, alright? So the type of mutation kita akan go one by one, alright? The type of mutation kita go for DNA level dulu lah, alright? So we go for the type of mutation is point mutation, right? Point mutation is this is the normal one, alright? The normal one, alright? I give you example macam glutamate ni tadi lah, alright? Glutamate ni tadi, alright? So the DNA of the normal DNA adalah CTC, lain semua sama Ini adalah codon, alright? The DNA sequence yang based on the codon lah, dia akan later will be transcribed into GAG lah kat sini, right? So this one will somehow protein will this RNA will be translated into glutamate, right? However, due to one point, right, due to the changes in A, right, sepatutnya T jadi A. What happen adalah bila dia jadi T, alright, dia akan bila dia jadi T, sepatutnya T dia jadi A, dia akan encode different type of amino acid, alright. So dia akan jadi GUG kat sini, right, when they're being transcribed. And then when they're being transcribed here, they will be translating this one as a value, right? So due to one point changes in the sequence of DNA, they affects the protein punya expression ataupun translate to value. So that is a point mutation. Dia berubah kat one particular DNA sequence, right? Perubahan, alright? So another type of a different type of mutation, I mean into detail of the point mutation ni, dia ada substitution lah, right? So mispairing ni biasanya mispairing, kita go to dekat yang kat bawah ni dulu, mispairing, right? So sepatutnya purine dengan pyrimidine, betul tak? Dia punya pair. However, tiba-tiba jadi purine dan purine. So should not be happening. Tapi tiba-tiba memang berlaku macam tu. It's mispairing, alright? So this is the huge error yang boleh berlaku lah, alright? Mispairing. Sepatutnya purine dan pyrimidine, tapi dia jadi purine dengan purine ataupun pyrimidine dengan pyrimidine. So this should not be happen. So this is one the thing yang abnormality yang kita boleh tengok during the mutation. What happen? Alright? When you talk about point mutation, these are two things yang kita always ingat. Transition is very simple. Purines tukar jadi pyrimidine ataupun pyrimidine tukar jadi purines. Sorry. Purines tukar, contoh purines A sepatutnya dia jadi G. Ataupun G sepatutnya dia jadi A. Pyrimidine pula sepatutnya C dia jadi T. Ataupun sepatutnya T dia jadi C. Alright? So this is the transition. However, transversion ni pula dia different. Alright? Dia different. Transition ni you are just want guys to have bear in mind. Dia adalah purine dengan purine, pyrimidine dengan pyrimidine. Alright? Purine dengan purine, pyrimidine dengan pyrimidine. Okay? So itu perubahannya. Tapi kalau transversion, dia berubah. Either dia jadi purine ataupun dia jadi pyrimidine. Alright? To make it simple for you guys, I prepare this one. Boleh nampak ke? Hello?
Boleh.
Nampak.
Alright, so this is transition yang color merah ni. Ini adalah purine either dia adenine ataupun dia berubah jadi guanine. So either one lah. Same goes with the pyrimidine. Alright, so cysteine ataupun thymine dia berubah. Alright, tapi kalau transversion dia berubah totally. Either jadi purine ke pyrimidine, pyrimidine ke purine. Ataupun dia berubah jadi purine ke pyrimidine. Dia boleh jadi dua lah. Alright, dia jadi A berubah jadi C ataupun A berubah jadi T. Right, ataupun G berubah jadi C ataupun G berubah jadi T. Ataupun G berubah jadi T. Dia either way around. Alright, so dia berubah dia punya properties. Alright, tapi kalau transition ni dia berubah dia punya base sahaja. Tapi still dalam group yang sama. Alright, kalau transversion dia berubah dari group yang berbeza. Alright, so this is a transition dengan transversion punya proses. Alright, so inilah yang kita tak nak berlaku lah. Due to one changes of that DNA sequence, it have huge impact on the protein punya function. Alright, so the next one we go to the frame shift lah. Alright, still under DNA level, we go to frame shift. Alright, what happened in frame shift sebenarnya? Alright, so frame shift ni either insertion ataupun deletion. Yang tadi ni adalah point mutation, dia substitution, dia bertukar. Right? Contohnya dia still berubah, dia still tiga codon and still 3 amino acid. Alright? Tapi kalau insertion ataupun deletion, right? Bila dia insert one particular DNA dekat dalam, dia dah menganjak the frame. Right? Remember, codon ni codon is in 3 DNA punya sequence, right? 3 DNA punya sequence, right? So this is one codon. Tapi bila kita ada one insertion macam C tiba-tiba muncul dekat sini, alright? So what happen dekat sini adalah frame dia dah berubah. Sepatutnya CTC, CTC, CTC. Tapi what happen dekat sini adalah CTC jadi CCT jadi CCT. So frame dia dah berubah. Alright, so this is what happened. Alright, sepatutnya glutamate dia jadi glutamate, glycine dan glycine. Dia berubah. So once effect dekat sini, dia berubah the whole sequence downstream dia. Dia berubah. Disebabkan kita dah ganggu dia punya frame of 3 codon tadi. Alright, same goes with deletion lah. Alright, bila kita delete one sequence, so frame dia pun dah berubah dah. So menjadi little bit shorter. Alright. So maknanya perubahan ni akan mengganggu downstream dia. Alright. So what happen? Dia ganggu a lot of the amino acid sequence downstream bila dia undergo translation punya proses. Alright, so remember this. In DNA level, dia ada point mutation dengan frame shift mutation. Point mutation ni normally is substitution lah. Perubahan one point, one DNA into another DNA lah. Alright. So what happen in frame shift pula, dia either insertion ataupun deletion. Bila dia masuk, dia bertambah, so frame dia akan berubah. Same goes with the delete. Bila dia dikurangkan, frame of the codon pun akan berubah. Alright? So, clear ke? Boleh faham ke ini? Ada yang kurang faham? Alright, it's good. Kalau tak faham, kurang faham, please let me know. Alright, thank you. So, okay. So, ni perubahan dia. Alright? So, bila dia point mutation, dia berubah one point. Kalau frame shift, dia berubah. Dia buang satu ataupun dia tambah satu. Dia merubahkan the whole sequence downstream. Right? So this is in DNA level. What happened in protein level pula? Right? In protein level pula, dia ada few lah ya, a few types. So the most common one, alright, the most common one yang kita tak nak berlaku lah sebenarnya. Right? In protein ni dia jadi nonsense punya mutation. What is nonsense mutation? Perubahan yang pada DNA, right? Dekat DNA ni menyebabkan perubahan kepada RNA menjadi stop codon. Remember stop codon? Alright, stop codon ni bila kita undergo translation process. Alright, undergo translation process, kita ada stop codon. Alright, stop codon ni UGA lah. So bila ada stop codon ni, dia akan memberhentikan the translation punya proses. Alright, which mean dia akan berhentikan amino acid punya ni lah, pembuatan amino acid in the during the process lah. Alright. So what happen, dia akan stop kat situ. Itu yang kita tak nak. So what happen sebenarnya yang berlaku kat situ? Bila kita stop kat situ, just imagine dia baru buat satu amino acid, tiba-tiba the next one jadi stop codon. Alright. Sepatutnya this function for more than 10 or 15 amino acid, for example lah, ni contoh. But tiba-tiba dia baru translate one codon punya sequence, tiba-tiba the next codon jadi stop codon. Alright, so maknanya this particular amino acid ataupun protein ni jadi tak lengkap. So it affects the whole protein punya function lah. Alright, so dia tak ada protein yang functional. So tiba-tiba jadi stop codon. Itu yang kita tak nak lah. So which mean that the changes of the DNA sequence that leads to the expression of stop codon. Alright. So bila dia stop codon, jadi stop lah. So memang that's it. Protein pun tak berlaku. Ataupun dia buat halfway. Contohnya protein tu sepatutnya dia ada amino acid ni sepatutnya ada 10. Tapi suddenly in the codon nombor 5 tiba-tiba jadi stop codon. So it's halfway to go. So it's a half protein punya structures. Non-functional protein lah jadi dia. Alright. So this is the thing yang kita tak nak lah. Nonsense mutation. Alright. What happened to other thing? Alright. So dia ada missense mutation. Alright. So missense mutation ni dia changes into a different type of amino acid. Remember the blood punya case tadi? Alright. They change from glutamine to valine. Alright. Alright, so they change of one amino acid. Dia jadi missense mutation. So missense mutation ni dia ada several jenis lah. Alright. So ni tadi nonsense. Nonsense ni jadi stop. Missense ni ada several jenis dan several condition. So what happen in this particular condition? Missense mutation dia ada tiga kategori lah. So first missense mutation ni jadi silent mutation. What silent mutation? Alright, silent mutation ni this is the thing yang saya sangat suka lah. Sebab saya pernah buat dulu and so on. A lot of things that I change. Alright, silent mutation ni codon dia berubah jadi similar amino acids. Remember ya, kan one codon can encode a few amino acids yang sama, betul tak? Alright, one codon can encode a few amino acids yang sama. What happen adalah when they change, the changes is into the similar amino acid. This is lysine. For example, glycine can be encoded by these four particular codon, CCA, CCG, CCT, dan CCC. Alright. So if they change A into G, they still encode glycine. If they change G into T pun still encode glycine juga. Alright, so this is the thing yang still ada lah. Yang ada semua dekat dalam ni. So apa yang ada, they will change into the glycine. Alright, so maknanya it's a silent mutation. The changes are with the similar amino acid. Okay, so these are the changes. So maknanya dia berubah pun dia tak mengganggu banyak the structure because it change into the similar amino acid changes. Alright, so dia tak mengganggu the whole things lah. Alright, so conservative pula. So what happened in conservative? Alright, they change the codon, alright, into a different amino acids, tapi properties dia still sama. Alright, so bear in mind if you try to find ataupun search, alright, amino acid yang dibuat ni, alright, amino acid yang dihasilkan, dia ada polar, non polar, ada size dia ada berbeza besar dan kecil, so alright. If they change into a different amino acid, but the property is the same, we call it a conservative mutation. Excuse me. Alright. So acidity dia sama, polarity dia pun sama. So normally changes is not really significant. Pun tak berubah sangat. Alright. Glutamine itu the aspartate acid. Alright. So the changes is not really effects. Alright. The codon change to a different amino acid with similar properties. Alright. So changes ni ini lagi memang tak change a lot lah because it change to the similar amino acid. But this change into a different type of amino acid, tapi property dia still sama. So no significant sangat lah. Biasanya alanine lah, ALU. Alright, ALU. So maknanya any changes to ALU ni dia tak mengganggu sangat. But ALU ni dia dah inert. Dia perubahan dia pun tak mengganggu apa-apa. Alright. Tapi non-conservative ni, codon change to different amino acid with non-similar properties. So this is the thing yang kita nampak dekat yang sel darah tadi. Right? Dia jadi valine. Valine ni, to be honest, valine ni, if you're not mistaken, dia non-polar dan dia punya size tu besar. So that's why when they change into different type ataupun different properties, right? So they affects a lot on that particular sequence. Right? So itulah yang berlaku dekat dekat yang sel darah tadi. Alright, it change to valine. Valine is a huge compound, huge amino acid, and the changes is affecting a lot. So this is non-conservative. Codon that change into different amino acid with non-similar properties. Right, this is what happened dekat yang sel darah. Right, tell you the acidity, polarity, size pun matters dekat dalam changes. Alright, so this is what happened in the missense mutation. Alright, so in protein dia ada dua type lah. Non-sense jadi stop codon terus. Right, missense pula dia ada 3 category. Silent mutation, conservative mutation dengan non-conservative mutation. Alright, so these are the changes. So another type is the last one is the chromosomal level punya changes lah. Alright, mutation is translocation lah. Alright, this translocation is gene from one chromosome swapped to different chromosome. This different from crossing over lah. Alright, crossing over ni different. Alright, I just want to highlight that's why color dia berbeza lah. Kalau color sama is sama. Right, so that's why crossing over will not be changed much. But this is different chromosome. They cross each other. So I give you kenapa I did mention here chromosome berbeza sebab you understand about human being dia ada 23 set of chromosome. So total ada 46 lah. Right, chromosome number 23 is the sex chromosome. Right, yang akan mengekspreskan jantina lah. Alright, so maknanya there's a lot of a different type of pair of chromosome ni for different type of functions. But for this one they cross. I mean dia dapatkan dia punya apa nama portion of the chromosome tu from different pair. Alright, so this is what happened. Gene from one chromosome swapped to different chromosome. So what happen bila dia ada swap benda tu, jadi berbeza. So maknanya information tu sepatutnya sama, tapi information tu terus berbeza. So that is what happen. Right? Ataupun inversion lah. Inversion ni dalam kromosom yang sama, tapi sepatutnya dia dekat arm, Q arm, jadi dekat P arm, bertukar. Right? This could happen juga. Right? This could happen. This could happen and this could happen as well in the chromosome. Two genes from the same chromosome switch place. Alright? Dia berubah. So sepatutnya dekat atas ni berbeza, dekat bawah ni pun berbeza. Alright? So this is what happen. Beza dengan crossing over. Crossing over dia dekat pair yang sama. Dia just tukar information sebelah-sebelah je. Alright? So that is what happen in the 3 different type ataupun kategori of mutation. Dekat DNA, dekat protein dengan dekat kromosom. Right? So source of mutagens ada banyak lah. Right? Mutagens from the chemicals, right? From the physical, boleh jadi gamma ray, heavy metal, arsenic and so on. Biological as well, right? Virus, transposon, bacteria, right? Carcinogen ni anything that which the potential getting cancerous lah, right? Tobacco boleh, UV light pun boleh, alright? So this is the thing yang tak nampak sangat dia punya carcinogenic punya properties, tapi it prolonged exposure can cause the mutation lah. Right? Tobacco dengan UV memang dekat cahaya matahari kita boleh dapat, tapi prolongation. Alright? So mutagen ni of course lah, we talk about mutagen, memang kita akan cakap chemical paling awal. Right? So this is the thing yang can affect. Right? Physical ni macam any source of light lah, gamma ray, x-ray, UV ray and so on. Heavy metal ni kadang-kadang kita tak perasan, but heavy metal exist, right? So I think if you guys know what happened in last time in the previous era in Japan, where they have the mercury punya leaking dekat dia punya sungai dan tasik, and how what happened adalah that mercury somehow being incorporated into the living things dekat sana, dekat ikan, alright? And when they eat the ikan, so they got the heavy metal. So what happened, that particular first generation tak ada masalah, the offspring of them having a lot of mutation punya issues, deformation of bodies and so on. Alright, so this is tak nampak, tapi this is the thing. People are heavily being study about this one. Kita tak nak ada heavy metal. I think you know lah, quite huge issue dekat sini lah, dekat Kuantan about bauxite and so on last time. But this is the part of the heavy metal as well. Right? So I guess this is all for me. Right? If you guys have any questions, do let me know. I'm so sorry to take your time. It's more than 20 or 30 minutes that I expect. Right? So any questions? Ada soalan?
Doktor,
Ya saya.
yang epistasis tadi,
Ya saya.
kan dia jadi defect dekat gene tu.
Yes.
Tapi dia tak semestinya jadi benda yang buruk lah kan?
Yes. That is very good question. Alright. So let me tell you one thing. Alright. When we talk about mutation, alright. So ni kita boleh consider as mutation lah. Dia berubah, hilang satu. Am I correct? Betul tak? Dia tak semestinya jadi benda buruk. Right? Mutation ni pula, if you go further lah later, you go for biotechnology and so on, right? So mutation ni can be a lot of things. It can become loss of function ataupun they can be gain of function. That's the thing yang kita selalu sebut lah. Mutation that loss of function, mutation that gain of function. Alright? Kalau the gain of function ni, itu yang kita lagi suka lah. Macam tadi, macam saya cakap, they can undergo drought tolerance. Right? So they might be loss of one of the genes yang unfunction, tapi they gain the ability untuk survive dekat keadaan yang out of the norm, kurang air. Itu dia punya capacity dan capability yang kita nak tengok. So to be honest, in agriculture punya area, alright, there are two types of studies lah, alright. One is molecular breeding, one is another conventional breeding. Alright, molecular breeding ni, such kind of this lah yang kita nak tengok sebenarnya. Apa yang berlaku? Contohnya one day awak pergi tanam 100 biji benih pokok cili. Alright, awak tanam semua. Sekali awak lupa nak siram, for example. Waktu lupa nak siram, sekali dalam 100 tu tiba-tiba awak tengok ada 2 ataupun 3 batang cili yang still survive. So what happen dekat situ awak boleh kaji balik. What happen sebenarnya? Kenapa dia boleh survive bila keadaan yang kurang air? So kita can go to see. Kita dah boleh kurangkan, maknanya dia ni boleh survive dalam keadaan kurang air. So bila kita cari genes yang related dengan drought tolerance ni, kita boleh tengok. There's a lot of genes yang evolve, kita boleh tengok satu-satu. Bila kita extract DNA of kita punya pokok cili tadi tu, kita tengok ada changes tak dekat dalam sequence dia. So if you got that, so that is what people do. People akan ambil that particular pokok, people akan try to breed them more untuk dapatkan cili yang ada resistance towards drought tolerance. Right, dia tak semestinya jadi bad ni lah, bad traits. Right, that is very good questions. Okay, another questions? Ada soalan lagi? Hello? Ada? Tak ada? Kalau tak ada, tak ada. Kalau tak ada, I would like to release you guys early so you can have a preparation for your laboratory this evening lah. Alright? So be punctual because kita nak dapatkan timing for each one of us lah untuk dapatkan the capacity dan capability untuk buat. Alright? So without further ado, I would like to end the session with tasbih kafarah for us. Please tengok dekat dalam ni lah ya, dalam ayat taklim, I will put your assignment inside there lah. Alright? Thank you so much for today. Assalamualaikum warahmatullahi wabarakatuh.